I'm just going to give people a minute to filter in. Okay, and while more people are filtering in, I'd like to say hello, everyone. Good morning, good afternoon, and good evening, wherever you are. Welcome to this week's iteration of the Long-Term Animal Research Seminar Series. We're going to continue this set of seminars with talks from Dr. Ann Hertel and Dr. Niels Dingemanse. And before I introduce our speakers, I have a few announcements. First, this will be our last live LTAR seminar. Next week, we'll have a special pre-recorded set of talks by Lizzie Speechley and Dr. Amanda Ridley. And so make sure to watch those via our YouTube channel. If you're participating via Zoom, for this talk, you should see a Q&A tab at the top or bottom of your screen. If you open that tab, you'll be able to type any questions that you may have, as well as see and upvote other people's questions. And at the end of the talk, we'll go through those questions, starting with the ones with the most votes. So we encourage you to use the Q&A tab. Lastly, recordings of all of our talks will be available on YouTube shortly after they conclude. So if you need to leave early or know others who are unable to attend live, this talk will be available for viewing and reviewing after they're both complete. All right, now that that business is attended to, I'm very happy to introduce our first speaker today, Dr. Ann Hertel. Ann received her PhD from the Norwegian University of Life Sciences, where she studied the effects of resource abundance on brown bear behavior and life history. She then completed postdocs on Senckenberg Biodiversity and Climate Research Center and the University of Southeast Norway, where she was funded by a prestigious Marie Curie Fellowship. Currently, Ann is a postdoc at the Ludwig Maximilians University Munich, where she's assessing the potential for humans to act as an evolutionary driver of individual variation. Ann's research program uses integrative methods to assess the causes and consequences of individual variation, and I particularly appreciate Ann's ability to use commonly collected data, such as biologging, to ask new questions about behavioral variation in the wild. So if you haven't read her work, I encourage you to check it out. I'm excited to welcome Ann today and hear more about behavior and dietary niches in a long-lived carnivore. So Ann, go ahead and take it away.
Yeah, thank you, Liz. Here we go. Yeah, thanks for the nice introduction. And thank you for having me today as Niels's pre-talk in a way. I joined Niels's lab last year, and I think we're having a really nice synergy despite working on very different species, especially also in size. And I hope, you're enjoying my talk today about how I can use movement data to study individual behavioral variation in brown bears. So really, I think my talk really fits into this seminar series because my work really hinges crucially on long-term data sets and long-term animal research. So I'm trying to delineate between and within individual variation in wildlife. And I'm especially interested in looking at over which timescales such variation is conserved and what are the sources of this variation. And at the end of the day, I'm trying to use this information about the extent of between individual variation and sources and to integrate that into population management of wildlife. Oops, there we go. Hence, my research is kind of at the interface of these three ecological fields of behavioral ecology, movement ecology, and wildlife ecology, using the literature, the hypothesis, and the concepts of behavioral ecology, using movement data, so primarily GPS locations of wildlife, to delineate behaviors in the wild, and then to integrate that into wildlife ecology, a field where there is so far relatively little appreciation for between individual variation in behavior. And so far, I've mainly used with the brown bear as my model system, but I've also done research on African elephants, and I'm currently also involved in work on Egyptian vultures. And to give you a little bit of an idea what kind of data I'm using and we're talking about here, so I've been involved with the Scandinavian brown bear research project since 2014, since the beginning of my PhD. But this project has been running for a lot longer. So they really established it in the mid 80s already, in the beginning of 90s. They streamlined protocols. And so we have a very nice long-term data set, multi-generational, where the project tries to follow females throughout their entire life and their offspring. And as you can see in the pictures, we're darting females in the spring from a helicopter, and we're equipping them with, in the early days, VHF radio collars, nowadays GPS collars. And we're really trying to get as much information on these individuals as possible. And just to give you an idea what such information can look like, this is Grivla. She's one of the bears that we've been able to follow the longest. She was born in 1993 to a female that was already monitored by the project. And in 2018, she was shot in the hunting season. So we have a 25-year-long data set for this individual bear. We know exactly when she reproduced and how often. That was 9 times for her. We captured and marked her offspring, all of her male and female offspring. And for some of her offspring, we even know their offspring again. So this gives us some nice pedigree and life history information on this bear. She was recaptured multiple times. And because we get as much information as possible, we know how her morphology developed throughout her lifetime. We're collecting hair samples, which can encode for things like the diet, and I'll come to that in a little bit later, or long-term stress hormones. And as I mentioned already, since 2004, bears are equipped with GPS collars, which take a spatial location for every hour of the day. And for Grivla in particular, we have a 12-year-long series where we can really delineate behaviors over the entire active season on very high detail. So with such a great data set, it's really possible to ask questions that go a little bit beyond just like the phenotypic, what do bears actually do? And there's 2 things that I want to present to you today. First of all, I want to talk about behavioral variation from movement data, and then second, about the determinants of dietary specialization. Yeah, so let's dive right in. So in this animation here, you're seeing 2 female brown bears. They're approximately the same age, both adults. They're solitary, no cubs at foot. And they were monitored over the exact same time period, so in August 2013. And despite the habitat in this landscape being quite homogeneous, these bears have very different space requirements. So the red one on the right-hand side travels over shorter daily distances, and she has a smaller home range. Meanwhile, the blue bear on the left-hand side has a home range that is more than 3 times larger than the one on the right-hand side. And she also travels over much longer daily distances. So I was interested in delineating what are really the time scales of this between individual variation. So is it mainly conserved on a seasonal basis? Is it conserved on an annual basis? Or are these true, one could say, intrinsic or personality aspects of these individuals that are repeatable over very long time scales, or ideally lifelong? So I've been focusing on 2 main behaviors. First of all, the daily diurnal activity, so how nocturnal or diurnal a bear is. And I delineated this behavior from accelerometer data, which are on board of the GPS radio collar. So here we get, for each 5-minute increment of the day, an acceleration into 2 orthogonal axes, from which we then can delineate how much of the bear's total activity during the 24-hour period was allocated during the daylight hours, here in yellow, or during the nighttime hours in blue. So we can construct an index, the diurnality index, which ranges from -1 to 1, where -1 would indicate a strictly nocturnal behavior, and 1 a strictly diurnal behavior. And the second behavior I looked at was daily movement distance, which I defined as the sum of all 24 daily steps. So essentially, I calculated the Euclidean distance between successive GPS relocation and summed them up over the 24-hour period. And I should say that both of these behaviors I consider as somewhat risk enhancing, because they both would increase a bear's likelihood to encounter humans on the landscape, which are in our system the only predator on brown bears, really. So for this data set, I collected data from 62 female bears, which in each year were monitored over 50 consecutive days. So I delineated these behaviors on each and every day over a 50-day period. And bears were monitored over 1 to 12 years, which allowed me to disentangle whether these behaviors are conserved over an annual or multi-annual basis. So the first aim in this paper was to see whether we have between individual variation in the mean expression of these two behaviors and over which timescales it was conserved. So how did I do that? I looked at my 62 individuals and my 50 repeated measures within 1 year. And I assessed whether a random intercept for bear ID, which would be mean behavior over multiple years, or a random intercept over a bear ID nested in a particular study year, so monitoring year, would explain between individual variation in mean behavior. But second, I was also interested whether individuals would differ in how consistent they actually are. So we call this predictability, and what it means is essentially that we're assuming that some individuals might have a greater residual variance or a greater standard deviation around their average behavioral type, whereas others might have a tighter distribution of their 50 days around their mean. So in this case, individual 2 and 4 would be highly predictable. So the mean would be better represented for them, whereas individual 1 and 3 have a higher standard deviation around their mean and are hence less predictable in their behavior. And last, I was interested whether these two would be correlated, so the mean behavior and how predictable an individual really was for this behavior. And first of all, I found that indeed both behaviors were repeatable, but on very different timescales. So diurnality was primarily or was highly conserved over the long term. So on this graph, you see on the left-hand side, the white individual has an average diurnality index of around minus 0.45. So it's rather nocturnal, whereas the whole gradient over the 62 individuals reaches towards a rather diurnal behavioral type with an average diurnality index of around 0.4. So within the same population, we have individuals that have a preference for being more nocturnal or being more diurnal, and this is preserved over multiple years. Meanwhile, movement was mostly conserved on an annual level. So between years, how far a bear would walk on average would vary quite a bit, but within a year, it was highly consistent. And just to give you an idea, our behavioral type for movement varied up to fourfold from individuals that moved an average of around 4 kilometers per day to individuals that moved an average of up to 12 kilometers per day. So that's ecologically also quite substantial. For predictability, I found similar results. So just to give you an idea, on the population level, the standard deviation around the mean for movement was 2.5 kilometers. However, on the individual level, that varied quite a bit. So we had individuals that were very predictable, that had a standard deviation around the mean of about 1 kilometer. So very, very predictable over a 50-day period in how far they walked every day. Whereas other individuals were quite variable. They had a standard deviation of around 5.6 kilometers around their average. So on some days they walked a lot, and in others they walked very little, and hence their mean was relatively unpredictable. Most importantly, the mean and the standard deviations, the predictability of these behaviors were highly correlated. In a sense, we had individuals that were quite nocturnal and or sedentary, so they didn't move a lot, and they were very predictable in those behaviors. Meanwhile, on the other end of the spectrum, we had diurnal and very mobile individuals, which were very variable and very unpredictable in their behavior. And I'm thinking that ecologically, there are two potential explanations for this functional correlation between predictability and behavioral type. And this is either that those bears that display these risky behaviors, so they are diurnal and they move a lot, they also meet more people in the forest. And in response to that, they change their behavior, which then leads to a more unpredictable behavior in our data set. Or alternatively, unpredictable behavior could be actually an adaptive strategy to avoid predators and in our population facilitate the persistence of risky behaviors such as diurnality or moving over long distances. As a second example of my research, I want to briefly talk about the determinants of dietary specialization. And similar to behaviors in the dietary literature, we talk about that we have a population niche for dietary items, and especially in generalists such as the brown bear, which feed on a lot of food items. This population niche could come because all the individuals in the population feed on the same food items, so they all have a very wide generalistic niche. Or alternatively, we have individual niche specializations where individuals within this population specialize on a small spectrum of dietary items. And on the population level, we have this very nice wide niche. And for the brown bear in particular, we really have a lot of diet items that they can forage on. So they are the poster child of a generalistic omnivore. They feed on grasses, on insects, on berries. They hunt moose cubs, but they also will scavenge on roadkills or on gut piles in the forest readily. And as I mentioned in the beginning, we collect these hair samples from each individual when we capture them. And we can use these hair samples to analyze stable isotopic signature in them, which encodes what the bear has eaten during the previous active season, or at least for so long, for when the hair was growing. So for this study, I used a multi-generational data set from 1993 to 2018, which comprised 500 hair samples of about 100 female and 100 male bears. And I delineated the trophic position, which is kind of an indicator of how carnivorous an individual is. And higher values indicate high carnivory, whereas lower values indicate a rather vegetarian diet. So there was two main questions that I was interested in. First of all, do we see individual variation in trophic position? And second, what are the determinants of such individual variation? To come right to the point, yes, we did see individual variation in trophic position, but that was clustered within the sexes. So generally, males were more carnivorous than females, but within the sexes, we saw high niche specialization. Now, in the second step, I was interested in where does this come from? So what determines whether a bear would be more carnivorous or more vegetarian? And I hypothesized that it could be either genetically driven, it could be due to spatial similarity on the landscape. So bears that live closer in space have access to similar resources and hence also have a more similar diet, or it could be due to maternal learning. However, this maternal learning, since we have such a long-lived animal, could fade over time. So I hypothesized that the effect of the mother on the trophic position of her offspring should be strongest in the first years after a family breakup. And that's exactly what we found. So in the first 2 years after the family separated, the offspring's trophic position was fairly similar to the one of the mother. So the mother's position was a good predictor of the daughter's position. But this effect faded over time. And in fact, after 5 years, there was no correlation anymore between offspring and mother. Second, we also found support for this idea that spatial similarity or spatial proximity would explain that individuals have a more similar diet, but no support for any genetic effects or permanent maternal effects. So overall, in summary, I'd like to say that I really value long-term monitoring data sets because I can use them to reveal the timescales over which individual specialization takes place, both in behavior, but also in diet specialization. And only the longitudinal data sets can allow us to really disentangle these between individual effects and to link them to life history traits that are ideally monitored over the lifetime of an individual. And it's only possible to isolate the drivers, the true drivers of this between individual and variation if we have multi-generational data sets, which for long-lived species is extremely rare and hence extremely valuable. So thank you for your attention. Thank you to Niels for having me in his lab, and to my previous partners and my funding organizations.
Great, thank you so much. I think Matthew has a question, so we'll start with him.
Sure, so I was really interested in this variation in diurnality within a population. It's pretty remarkable. And so I'm curious whether you have any data that can kind of let you estimate levels of human activity that the different bears might experience to see whether that's actually predicting their diurnality.
Yeah, so we have proxies. We have spatial proxies. So we have road densities and we have human density, so build-up density. And surprisingly, none of these explain any variation. So we would expect that bears that live closer to humans would be more nocturnal. But I guess the thing is that in our study area, there is relatively little permanent human density. There is relatively high so there's people coming in for hiking and for berry picking, and so recreational activity, but no permanent activity, and that's very hard to measure. So ideally, what we would have would be something like traffic volume on roads or on trails, but unfortunately, we don't have that.
Thanks.
Cool. And I also have questions. I'm curious if I was struck by the variation in movement distance in the inter-individual variation. And I'm curious if you know if it correlates with other phenotypes like body size or aggressiveness or any other things.
Yeah, so it does correlate with the diurnality. So diurnality, these two are a behavioral syndrome, so to say. We haven't really correlated it to morphology yet. That is part of my current project to try to correlate it to morphology, to stress hormones, but we don't know that yet, sorry.
No, cool. I'm looking forward to hearing more about it in the future then. Great. Thank you so much. I think we'll switch gears now, and I'm very pleased to introduce our second speaker, Dr. Niels Dingemanse. Niels is a professor of behavioral ecology at the Ludwig-Maximilians University Munich. Research in his lab addresses the causes and consequences of behavioral strategies using experiments and observational data in both the lab and the field. His work integrates ideas from behavioral ecology, life history, and quantitative genetics theory to ask when trait integration between life history and behavioral strategies, for example, is favored in wild populations. Niels' lab is also at the forefront of developing statistical tools to quantify variation at multiple levels and has authored seminal papers on partitioning individual variation in behavior. So today I'm thrilled to hear him talk more about the value of long-term data for assessing animal personality. With that, go ahead and take it away.
Yeah, I would like to start with thanking both of you for organizing this wonderful program, because this is really, I think, a valuable way for people around the world to be able to enjoy interesting contemporary science. So many congratulations on that. So what I'm going to talk about today is animal personality. So I'm going to follow up some of Anna's introduction today, and I'm going to focus on one species in particular. This is the gray-tit. And you see the gray-tit in the upper left corner here. We've also studied many other animals, but those are not species where we have these nice longitudinal studies, and therefore I chose the gray-tits because this is a very nice example of the value of longitudinal studies. So I've been around now for a few years, and when I started my PhD, there was an era in behavioral ecology that some characterize as being the tyranny of the golden mean, which sort of implies that people were very much obsessed or interested in how the average animal in the population would behave on average, And there was very little appreciation for the notion that there might also be some individual variation. And if it was there, that mattered in any way, okay? And then at some point, there were people like me and other people like NDC and Denny Real and Alison Bell and many other folks that were working on a whole diversity of species that were actually demonstrating, well, individuals from the same population are actually quite different. And this is very nicely illustrated here by this Andy Warhol type of cartoon. This is the front page of a special issue that came out in 2010, which is already over 10 years ago. And that pushed the field really forward by moving us beyond this tyranny of the golden mean. And one way by which we achieved doing that, and maybe one picture in which we achieved that, and that's actually quite cool, this sort of moving beyond and breaking through boundaries. Here you see a picture of two of my former PhD students, Erica and Robin. Erica was a co-PhD student by Bart Kempenaars and other people at the Max Planck Institute for Ornithology. Okay, so the way that we did that, right, is by viewing behavior in terms of reaction norms. And that was relatively novel at the time. And I will illustrate that here. So imagine that each individual in the population is shown by a line, right? Then in the left-hand panel, in panel A, we see a situation where everyone is the same. So all the lines overlap and everyone is plastic in their behavior, responding to the environment. So the reaction norm is non-horizontal. And this was sort of one of the common paradigms that behavior is superplastic. But we pointed out actually through a number of studies that individuals might also be very different in their behavior. So that's panel B here, where individuals have different intercepts for their reaction norms. And these differences between individuals are in this case maintained across time or different contexts. And we also pointed out through numbers of studies that both variation within individuals and variation between individuals could exist at the same time. And that was an interesting insight that was very nicely visualized by thinking in terms of these reaction norms. So here you see this panel C that shows that individuals might differ in their average behavior, how they respond in, say, the average environment, but individuals are at the same time plastic in their behavior. And these sort of individual differences in behavior, these consistent individual differences despite behavioral flexibility that behavior shows, this is what we call animal personality. Okay, so now what I'm going to do today is talk primarily about aggressiveness, and I'd like to start with a nice illustration that has been made by James Munro. And he's an artist that helped illustrate a really nice children's book. And that children's book was made by Tiffany Taylor some years ago. And this really illustrates very nicely what we study in a nutshell. So you see different types of braidheads here. So we have a bald guy, for example, and we have some shyer individuals. There's also some assortative mating. The bolder guys are mated with the bolder guys and the shyer with the shy guys. And what we see here is that there's not just variation in behavior, there's also variation in life history. So we see that these bolder animals at this point in time produce very fat, juicy chicks. And they also live in a very different type of environment, as you can see, because these bald cratids inhabit here a nice oak tree with lots of juicy caterpillars, right? So this sort of sets the scene, really, that there can be variation in fitness that is linked to these different behavioral types. Okay, so what I will talk about today is do gratids in the wild really differ in their personality? So I'll talk a little bit about that. We've given it away already. The answer is yes. And then is this variation on the selection? And that's, of course, difficult to study. And for that, you need longitudinal studies to quantify both behavior as well as selection. So how well these animals do in different environments. And that's not so easy, actually. And then the third question is, can we understand how this variation is maintained? Why is this variation actually there? And our view is that by understanding individual variation and by quantifying variation in the patterns of selection, we hope that we can solve this third question, okay? Now, this requires long-term studies, and I think in this paper by Matthew and Liz, this is very nicely summarized, and I just repeat here a couple of points that really apply to our line of work. Okay, so we require longitudinal studies to distinguish within from among level processes, particularly within from among individual variation in behavior, and already talked about that at some length. We can also use these types of longitudinal studies to better quantify integrative types of, or even latent characters. So characters are, you could say, aspects of an individual that are not expressed at every observation, at every occasion, but that we can actually back quantify if we have lots of repeated measures. So we can, for example, work out how does an animal behave on average. That's not something that's a phenotype that's expressed, that is actually a latent character, right? For that, we require longitudinal studies. And of course, if we do such studies in the wild, we will find variation. And one of the aims really here is to quantify the extent of ecological variation and then ask questions about how that maps onto patterns of selection. And of course, longitudinal studies will also allow us to estimate via these pedigrees that Anne showed, some pedigrees from parents to offspring. It allows us to quantify inheritance. I'm not going to talk much about that today, but it will also allow us to quantify variation in the patterns of selection and link those to those ecological factors. And in that way, we should ultimately be able to connect both ecological and evolutionary processes. So that's the idea. So what do we know about great-tits? So much of the audience might not know this bird. The great-tit is a very common passerine bird. It breeds in nest boxes throughout Europe. If you put up a nest box in your garden, the great-tit will breed there. On average, they lay about 8 eggs. Most birds live for a few years. The annual survival is about 50%. So you can imagine they don't live very long. Some birds live for 2 or 3 years, but many animals don't actually survive for a single year. Now, what's interesting about these great-tits is that you can actually also keep them under standardized conditions. This is not so easy, but there's some labs specialized in doing that. And one of the earliest studies on great-tit personalities, this was work done by Piet Drent and Monica Verbeek, and other people like Wiepkema. They took animals into the laboratory as nestlings and then raised them by hands. This is a massive amount of work. You can imagine that you have to feed these little nestlings by hand. But the nice thing about that is that you can then really keep them under standardized conditions. They did that, and the aim was really to create a common environment to then screen these animals for a whole range of behavioral tests. And the reason for that was that there was previous work on rats and mice that showed that individuals had really different, what we now call coping styles. So different ways of coping with internal and external environmental stimuli. And the easiest way to think about this is that some individuals are very much internally organized. They don't really look like, don't look at the outside world very much. They're internally driven. But other animals might actually adjust their behavior to the environment, right? So sort of flexible and inflexible types of animals. So this work was sort of based, right, on that body of literature from the rodents that was, of course, collected primarily in the laboratory. And Monica Verbeek, she was one of the first PhD students working on this project. She then hand-raised these animals and then tested them for all kinds of interesting tests. And one was an exploration test in a novel environment. You see that here, where animals are released into a novel room that they've never seen. And then they are basically asked to explore this environment. And animals differ very much in how they do that, and that behavior is also repeatable. She also exposed animals to novel objects that they for sure would never have seen. For example, this pink panther. We cannot do this test anymore because this particular model of pink panther doesn't exist anymore. Okay, so there's some issues with study replication. And she showed that individuals were also repeatable in that aspect. And the interesting thing was animals that are very quickly or boldly approaching this type of object, like this pink panther, also boldly approach other types of novel objects like a battery, for example. Okay, so you already see that there's some structure in the behavior. And then these animals that were explorative and bold, there were also animals that were very aggressive in pairwise confrontations. And nowadays, so this was work done in the 90s, so quite a while ago, nowadays we are very familiar with the notion that across a large range of taxa, ranging from spiders to insects to birds to fish, there is what we call the aggressiveness boldness syndrome. So there are animals that are more aggressive, more bold, explorative, more active compared to others that are less so. Okay, but that at the time was actually very novel finding. Now, that work was then, of course, followed up by work asking questions about, is this actually heritable variation? And her advisor, Piet Trent, he was working at the Netherlands Institute for Ecology, followed that up and performed selection line experiments. So you can imagine you pair the fastest ones, the boldest ones with the boldest ones, and the shyest ones with the shyest ones, and you do that for a number of generations. And then if you see this response to selection, what you see here in the actual data, then that implies that this behavior is, of course, heritable. Otherwise, you would not see such a response. And from the slope of these lines, you can then calculate the heritability in the broadest sense. And what they were finding, which is quite remarkable, is that the broad sense heritability was about 50%. And that means that half of the variation that we see in behavior is due to combines the effects of genes. Okay, and that at the time was very interesting and also unexpected. This is at the turn of the century. Then other work followed that up. Actually, work by Kees van Oers. I will show a picture of him in the next slides. And he showed that that all these different aspects of behavior were essentially both heritable, but also genetically integrated, genetically correlated, probably not due to pleiotropic effect of genes, but due to linkage disequilibrium. So we have learned over the past few years quite a bit about not just patterns of selection, but also the genetic architecture. And check out his work for more recent advances there, also in the context of methylation, for example. We won't talk much more about it. Okay, so in the late 90s, a research program was started, and here you see a picture of Kees, and also myself in little cartoon form in this children's book that I just showed you before. But there were other PhD students. There was also Claudio Carrera and postdoc Christian Bots. He works on climate change now, Christian. And we were together in this program, and it was very unique because this was a program that was a collaboration between many different Dutch universities with scientists that had very different bits of expertise. So this was really actually quite an amazing type of program to be part of as a PhD student. And my job there was to go back into the wild, right? So remember, I talked about laboratory-raised wild gradients. Of course, we cannot measure patterns of selection if we don't go back to the wild. And so we then design tests on gradients. Here you see a wild gradient in the laboratory. And the way that we approach this sort of problem is that we said, okay, let's catch animals in the wild. We have a picture here of an animal that's captured in what we call a mesh net. Okay, so here's an animal hanging in the net. You also see that it's ringed, for example, so that we can follow it for its life. We captured these animals, kept them overnight, and then early morning, one by one, they went into this exploration room to be tested for their behavior. Then they were released back into the wilds. Okay, if they would survive for a year, we would catch them again to the same procedure. Okay, so here you see just a plot of the variation. And what this shows is that even though in much of my presentation and presentations of others that you might hear about animal personality, we often talk about types, right? Bold and shy types of animals. Really, we're talking here about continuous variation typically. And this is actually quite a large sample size. It's a couple of hundred of birds, maybe 1,000 birds. And you really nicely see this follows a Gaussian distribution. Okay. Now, in the late 1990s, we started doing these types of tests, and we had no idea whether wild gradients would also be different, consistently different in their behavior. So that was actually quite exciting. I was also, of course, quite nervous as a PhD student when I did all these recaptures to see, Are these individuals just completely plastic, or is there some signal of individuality? And this was actually the case. And just to put this back into this reactional perspective, basically this is how this variation looks like. There's individual variation in behavior, this exploratory behavior. This behavior is also heritable. But at the same time, of course, these individuals are very plastic. They're responding to changes, for example, in environmental conditions like seasonality. They respond to seasonality. They also respond to how long it has been ago that they last were in this environment. So there's a novelty aspect also that weighs into this behavior, as you might expect. Okay, but the question that then came to our mind later on is, of course, is this something that just characterizes our pet population, right? We all have our pet populations that we are monitoring maybe forever, but the patterns that we find there, are they specific to that one population, or is this more generally applicable? And only if the latter is the case, then we can actually generalize. And I would say we would actually have learned something about how we might predict the world in biological terms. And luckily, some years later, a number of other research groups also started to measure this type of behavior. And here you see pictures of some of the people leading these studies, John Quinn and Ben Sheldon, Erik Matthijssen, he was in Antwerpen, John and Ben were working in Buiten Woods, and then Joost Timbergen was working in another population in the Netherlands. This Westerheide population is where I did my PhD. Now, what was interesting about these studies that these other researchers were doing is that they had duplicated our approach entirely. Okay, so here we see a setup that everyone used. We started by doing these exploration tests arbitrarily somehow in a novel environment room that was exactly 2 by 2.4 meters, because those were the dimensions of a laboratory room available at the Netherlands Institute for Ecology. And then throughout Europe, this type of room was actually replicated in exactly these dimensions, so that we can do more or less the exact replications, right? So that was quite nice, because with these identical setup, we have then better means to also compare these different populations. Okay, now, so people have done lots and lots of tests over the years, and here you see again these reaction plots. Here we have a line representing an individual. This is their best linear unbiased predictor, so we can control for all kinds of effects and sort of estimate what the individual is, right? On average, what we expect it to be during its first test, but also during a repeat test. That is an important distinction whether it's a first or repeat test, because this is a response to novelty. And what we see here is that there is just a lot of variation, right? Lots of variation. So how do we make sense of that? And how can we even compare that across populations, right? So if you stare a little bit at this, you might think, okay, you know, it looks all very similar, or maybe they're quite different. So how do we look at this actually? Now, there are standardized ways of doing this. And the approaches that we used are approaches that are really borrowed from quantitative genetics that imply the usage of variance partitioning approaches. So let me talk you through that a little bit. So how do we quantify this individuality? The way that we do this is that we take lots of repeated measures of the same individual, and then essentially for this individual, we can make a little density distribution, right? So imagine that we have this sort of frequency distribution of the blue individual and also of the green individual, right? And what we see here already is that these two individuals are plastic because they're not always showing the same type of behavioral value, but at the same time, their mean values also differ, right? And this is how a whole population of data points might look like. So this is data on aggressiveness, actually, that we collected on the gradients some years ago. So there's variation within the individual. This is how each observation deviates from the individual's mean. That's called within individual variance. But there's also variation between individuals because individuals differ in their average value. So the variance among the means is the among individual or the individual variation. And we have this residual within individual variation. We can add these two things up. And that's then, of course, the total phenotypic variation in the sample. And now we get to how do we quantify this individual variation? We quantify that by dividing the individual variance by the total. That's called repeatability. And Anne was already showing these metrics in the context of the bear movement behavior. There are problems with the repeatability because this is a fraction, so it doesn't tell you maybe so much as we might think. So for those of you interested in understanding individual variation, check out recent papers that talk about the value of repeatability versus other types of metrics. But this is a standardized metric that is widely used actually to understand the extent of individual variation. So back to the graded. We quantified then this repeatability because we collected repeated measures in our wild graded populations. In the Westerheide population, where I did my PhD, repeatability was about 40%. So that's interesting by itself, because what it actually demonstrates is that even though before we were often thinking, well, behavior is super plastic, animals are flexibly adjusting their behavior, 40% of the variation in behavior is due to differences between individuals. This is actually quite substantial. Right, so that was actually quite an interesting insight at that time. Now, in other populations, how do these patterns look like? In the Lauwersmeer, same thing, repeatability about 40%. In the Boshoek population in Belgium, a little bit lower, but sort of in the same range. Here you see the 95% credible intervals. White and Wood, the same thing. So what this really means is that if you sample a population of gradeds, you will find that individuals are actually repeatable. They're different in their behavior. And you will also find that these differences are actually very similar across different populations. So maybe they're they're the same types of pressures, ecological and selection pressures, acting on these animals that lead to these levels of behavioral variation. Now, as Liz was already alluding to, I'm no longer in the Netherlands. I moved at some point to Munich, right, which is more in the southeast of this map, in southern Germany, near the Alps. And there we've also continued some of these tests. And this is work by Alexia Michel. She just defended her PhD. And she was also finding for our population the same type of pattern. So that's quite nice, I think, because often people say, why would you ever replicate something that we know? Only when you replicate, you know whether something is actually predictable across populations. So I think that's actually a very important lesson from this type of exercise. Okay, now let's put this a little bit in a wider perspective. So if we look at hormones, metabolism, and behavior, there have been meta-analysis done of the studies that collected repeated measures. And this is work that I'm presenting here by postdoc Benedict Holtman, who is now a postdoc in my lab. He did this work with Shinichi Nakagawa, and he showed in this meta-analysis that basically all kinds of different types of behaviors in the context of parental care or anti-predator behavior or social interactions, typically behavioral traits have this repeatability of between 30 and 40%, much higher than hormonal traits. You can imagine that hormones can go in all kinds of directions very quickly, lots of measurement error too, maybe, and sort of similar to metabolic traits, right? So that's what we know now, quite a few years later. So this is a paper that came out in 2017. So individuals are different in suites of correlated traits across the animal kingdom. Okay. One of the interesting problems, of course, is the question of whether those differences in behavior are actually genuine or not. So is this actually repeatability or pseudo-repeatability? Pseudo-repeatability implies that it's not what it seems. Maybe these individuals are not different after all. And this is nicely illustrated here. You see bird A, we measure bird A twice it looks to its left, okay? And then we measure bird B twice it looks to its right. Okay, and you would say, well, those birds are consistently different in their behavior. The behavior is highly repeatable. But now we might realize that in the middle, there's actually a predator. And the reason that this bird is looking to the left is because the predator is at its left. And the reason that this bird is looking at its right is that the predator is at its right. So if we would turn the directions of the predators, we might actually see that the individuals would look at a different side. And therefore, we might also conclude that they are not repeatable after all. They might just be fully plastic. So to understand the extent to which this individual variation is genuine, you could say, what we have to do is do manipulations of the environment and then measure the behavior. And this is what we've done through a number of studies. So I will just show you one example of that. This is the study area that we use, those are 12 different nest box plots, each with 50 nest boxes. And the nice thing about this setup, those were 12 different plots that I've studied for about 10 years when I was at the Max Planck Institute for Ornithology. That's now a number of years ago. This was work done in collaboration also with Bart Kempenaars, who is a director there at the Institute. And what you see here is different colors because we manipulated these plots in different ways. So the ones in yellow are plots where throughout early spring, we were broadcasting the sounds of blackbirds. And the blackbird is a songbird that we have here. It's sort of similar to the American robin, although it looks it's completely black. Okay, so it's sort of very different at the same time. Very beautiful song. In other plots, we were broadcasting sounds of the main predator in the system, and this is the sparrowhawk. So the sparrowhawk is a predator that is sort of an ambush predator, and it would hunt small passerine birds like these gray tits. So we manipulated the environment in this way, and the way that we proceeded is with a multi-year partial crossover design. Now, what does this mean? So in a single year, a plot was manipulated in one way. So we give the birds the impression that they're predators or that there are no predators. And half of the plots would switch the treatment to next year, but the other half would not, right? And because some of the plots are not switching treatment, you can calculate where the birds are repeatable in a predation context. And you can also calculate where the birds are repeatable when there are no predators around from other plots where we have another treatment repeatedly across many years. But from the plots that are changing, either from one context to the next or vice versa, we can then ask questions about whether there is cross-context consistency in their behavior. So where the birds that are relatively bold, if we now completely change the environment, and this is a major manipulation of their environment, do we actually see that their behavior will also change or not? So here we see a picture of Robin, who carried around 1,000 kilos of batteries to do these types of experiments, really completely crazy. Here you see our Fox Pro speaker. And of course, we meet lots of people, so our students at work as well. Pretty cool, right? So really complicated types of experiments. This is the general idea. So if these animals have personalities, if they're consistently different in their behavior, we expect that we have this cross-context consistency, okay, across the context of low and high predation risk. We can actually, the most easiest way to quantify this is by looking at the correlation between the context. So if you read the paper, you'll see that the stats is a bit complicated, but we'll just not talk about that actually now. We're just going to show the correlation between how an animal behaves in one versus the other context. And we're expecting that to be tight if they have personalities. And this was actually the case. And so one thing to keep in mind is that the repeatability of those behaviors in each context is actually relatively moderate, about 0.4, but the cross-context correlation, which looks at the correlation between the mean values, can actually be very high. And this is something that is a bit counterintuitive, but maybe if you're interested in these types of questions, have a look at that paper and you'll see that there's some complex math behind that. But what it actually basically shows is that yes, these animals do actually have these genuine personalities. This is not just pseudo personality. There are really these real differences. Okay, now how is this variation maintained? That is now the next question. We've established a little bit, right, that there are differences between these types. We also know that they differ in aggressiveness in the wild and all kinds of aspects of foraging behavior and so on, but I'm not going to talk about that. The different types of ideas. One idea is that individuals resolve life history trade-off differently. Another idea is that because there are these genetic correlation structures in behaviors, that if there are selection pressures going in different directions on the different components of behaviors, that we cannot really evolve this sort of set of behaviors easily. So there might be strong constraints on the evolution of behavior. And we have done some review work suggesting that is the case. Some newer work looks at social environments. So the idea that if you have different personality types, because you interact with different types of animals, this might also be a mechanism to maintain this variation. I'm not going to talk about that much further. What I'm going to focus on particularly today, also in the context of the long-term studies, is a notion that we have fluctuating selection. We'll briefly touch upon this notion now that individuals might have alternative ways to resolve trade-offs or alternative ways to achieve, to maximize their fitness, right? Before we go to talk about fluctuating slope. Now let's go back briefly to this experiment. So where we have animals exposed to low and high predation risk. What I show here is a plot that shows the exploratory score on the x-axis, so an aspect of their personality, and then on the y-axis the lay date. The lay date is the date in the year that they start laying. So some birds are very early, they start laying early April, and some birds are very late, which means a few weeks later. And this really matters for them. So birds that lay early do much better than birds that lay late, for example, right? So this is a major life history trait. And what we see here in this graph is that when the perception of predation risk is that there are no predators around, low predation risk, the fast explorers breed relatively late in the year compared to the slow explorers. But this pattern actually reverses when the pattern of predation risk suggests that there are predators around. And so this means that fast explorers actually shift forward and slow explorers actually shift backward in the season. So this must have fitness consequences because laying early is so important. Now we've done a path analysis to look at this. And this suggests actually that we're talking here about alternative reproductive strategies because these types of animals, in whatever environment you put them, in this case, they have equal reproductive success. I will talk you through this very briefly. So what we see here is three boxes: exploratory behavior, lay date, and clutch size. And so what we see here is that explorative animals, they breed later in a control condition. And as I already revealed, they therefore produce fewer eggs. But what we also see at the same time, you can do this with the path analysis, you can look at indirect effect. This is an indirect effect, but also at direct effects. What we're seeing is that when these two types of birds breed exactly at the same time, so when we control for lay date, the faster explorers produce more eggs. So fast explorers produce more eggs, but they also breed a little bit late, and therefore they produce fewer eggs. Overall, they produce actually, if you do the calculation, the same amount of eggs. So even though they lay different times in the year, that has fitness consequences, they have the same reproductive success. Now in the predation risk environment, we find actually the same type of pattern, that they have equal reproductive success. The faster explorers now shift forward, they lay early in the year, therefore they actually produce more eggs. But at the same time, we see that this residual effect, that fast explorers simply produce more eggs, is actually diminished compared to what we see in the control environment. And this probably has to do that they're wasting, you could say, time and energy scanning for predators and so on. But overall, the pattern here is that, again, these two types have different reproductive success. Here we see the cross-context correlation that I just showed you in the visualized on the outer screen. So animals that are explorative in one context are also explorative in the other context. And at the same time, we see that animals that have a high clutch size in one context also have that in another context. But the lay date is actually individually plastic. So the way by which lay date is changing differs between different types of birds. Okay. Now, one of the, so this implies that there might be some processes by which two types of birds, or this variation in types, might come with equal reproductive success consequences. I'd like to now talk about this fluctuating selection type of idea, and this is because much of the theoretical work in this area, so this is work by people like Max Wolf and Vanja Wijsing, NDC, for example, here on the picture, have done lots of work in that area. They imply using a behavior ecology paradigm that the optimal behavior depends on state. So that's visualized here in this graph, like in evolutionary ecologist terms. I'm more evolutionary ecologist than behavior ecologist, I always think. So here we see behavior and the environmental state, the state of the environment, and fitness. And what we can see here essentially is that in different states of the environment, a different type of behavior is actually favored. So this is nothing more than saying that change your behavior when you are faced with a different type of environment. Or said differently, maybe a different personality type is favored in different environmental conditions. This really implies that in different environmental conditions, we have different selection pressures. This implies that there is fluctuating selection acting on these personalities in a heterogeneous environment. And this is what we've tested in sort of the broadest sense of the words. Okay. Now, fluctuating selection can come about in many different ways, actually. Now, we'll briefly talk about it. Okay. So we have variation in selection pressures and their consequences of that. So there can be macro spatial variation in selection. And you can imagine that we have, for example, forests full with particular types of trees that deliver like particular types of food to animals, where there is selection for a particular type of bird compared to other types of forest. Okay. This can, of course, also act at a micro scale, right, within single population, right, the patches of different types of habitats. And the consequences, of course, over evolutionary time, when we're talking about heritable types of behaviors, that there is selection for local adaptation within these patches. So there's a particular type favored, and we are expecting local adaptation. Now, this might not always be the case. For example, if we're talking about a dispersive species, a species that's highly dispersive, like aggressiveness, actually, if we have different peaks, actually, in terms of fitness optima, then different types might be favored in different places, but because there is dispersal, abundant dispersal, this might then also help us understand why the variation actually is maintained in these populations, right? So looking at spatial variation in selection, that's an interesting thing to do. Now, there can also be temporal variation in selection. This means that within the same population, selection changes from one time point to the next. For example, because there's variation in predation risk or also food availability that also varies temporally within populations. And of course, these patterns can interact. So imagine, for example, that in areas where there are beech trees, so in North America, you also have the beech trees, right? They're masting every now and then, every so many years they're masting, there's lots of food, and that then changes the patterns of selection at the places where these beech trees are, but in other populations where the beech trees are not around, patterns of selection are not changed, right? So the idea is then that there are often also interactions between spatial and temporal variation in selection, right? So those types of patterns of selection we've also quantified. So whether the patterns of temporal variation differ between populations and also between habitats within populations. Okay. Now, temporal variation would typically reduce the speed of local adaptation, so there might be overall stabilizing selection, but if we have lots of fluctuating selection, then this sort of dilutes that pattern a little bit. But in age-structured populations, theoreticians imply that also this form of variation in selection can help maintain variation. Okay. Now, so what we've done in our study, we continued this successful collaboration with the people all around Western Europe, so in the UK, Belgium, the Netherlands, and also in Europe. Here you see a picture that shows that there's tremendous spatial variation also in environmental conditions. This is this beech tree that provides winter food for the grey tits every 3 years, then not, and then every 3 years, and so on. And I should say, these types of studies, if you are an early career researcher and you're wondering, should I collaborate? Yeah, collaborate. Collaboration is a very good thing. You can do things that you cannot do alone, really. So try it, do it. You'll make friends, and you will also benefit from that jointly scientifically. You can do things you otherwise can't do. So here we see now a setup of our 5 populations. This is how they're distributed over Western Europe. What is nice about these populations is that in some populations we also have different plots. So we have different micro spatial variation also. In Lauwersmeer, for example, in Zeewiesen, we've already seen that in Western Germany, southern Germany, and also in Belgium, the population by Erik Matthijssen. Other population, there's just a single nest box population, right? So we have like large scale spatial variation, but we can also zoom in a little bit and look at spatial variation at the micro scale. So we have data for 5 populations, 35 of these little nest box plots. So that's quite a lot of data. 18 years of data. Longer study is done in the Westerheide that has been continued by Kees van Oers. And we're looking at thousands of birds that are assayed for that personality over the last 20 years or so. Completely crazy, of course, but not just measured for their behavior, also measured for their reproductive success. How many eggs do they lay? How many offspring do they produce? And so forth and so on. Okay, now, bottom line, there's actually no selection on this behavior overall. So that was a bit of a surprise. So you'll see that here, if you look at linear selection gradients, that's this beta here, we see that when we look at survival, the selection gradient is completely flat. So there's absolutely no effect of your exploiter behavior on how well you survive on average. Also not on recruitment selection. Recruitment selection is a measure that we often take. That's a number of offspring that you produce that also recruit and become breeders themselves. So it's sort of a nice fitness metric. And we can also add the two up in some way. We can have an integrative fitness type of metric. And all of these show, yeah, there is just no selection overall. So in the average population, in the average year, in the average plot, there's no selection. So that was interesting, actually, a bit unexpected also, but that was very interesting. What it does imply is that some of the common ideas that have been around in the literature for some years are probably invalid. And one of the ideas is that there is selection for one type, so eroding variation, but at the same time there are mutations coming in, and therefore we have sort of mutation selection balance. That idea doesn't really seem to apply here because there is no selection overall. And also in the context of human personality, other studies, for example, those reviewed by Lars Penck and others, they imply also that this is not the case in humans, right? So that's actually quite interesting what you can get from null results as well. Okay, but at the same time, we find very strong fluctuating selection. Okay, so let's look at that. So here we have the picture that is shown. So every dot here is one of these little study plots in a particular year. Okay, so that's these dots. What we see here is our 35 study plots. They are in different populations in different countries. And here we have the linear selection gradients. Okay, we can also do other types of selection gradients that doesn't actually matter for the interpretation. Okay, just believe me, that's true, right? That's in the paper, of course. What we see here is that there's variation across different plots. We also see that there's some variation across the different populations, and we also see that within a single population there is variation in the patterns of selection. Sometimes positive, sometimes negative. So positive means the fast explorers do better. And negative means the fast explorers do worse. And what we see overall, it's zero, actually. Overall, there is no selection, right? So overall, the average bird does best, you could say, but there is variation in selection, okay? And so positive values, fast explorers do well, negative values, slower explorers do well, okay. Now, the question, of course, is what explains this variation in selection? And I think because we have these longitudinal studies, and we also have many different replicates, you could say, different populations investigated by different researchers, we can actually partition this variation in selection, which is a bit of a crazy idea, right? So we have these selection gradients, that's the slope of this relationship between the standardized trait value and fitness, and this slope varies a lot, right? And maybe in space and time. So what is the relative contribution of spatial versus temporal variation, for example? Okay, let's look at that. Now, what I'm going to show you in a minute, so we're now back to thinking about how well the gradients do, right, in different conditions. We're finding in this study that there is strong population variation in patterns of selection. This is one way to illustrate that, right? In some populations, faster, bolder guys do well, in other populations, the other way around. We're also finding that if we zoom into different habitats, we find the same types of patterns, right? That bolder types do better in one habitat and shy types in another habitat. And at the same time, we see that across Europe, across time, because we monitor the same years across these populations, we are finding that in some years, particular types of birds do better, in other years, other types of birds. And then there are, of course, these interactions. Okay. Now I'll show you the numbers, right? So think about this as a pie, right? We have lots of variation in selection pressures. How much is attributable to which source of variation, okay? Now what we're finding is that macrospatial variation. So this is variation in patterns of selection among the West European population that accounts for most of the variation. Now, this is super interesting because many of us, of course, will always only look at our own population. So this pattern of selection, this variation across population, you would just never know about, right? So this shows this value of working together. At the same time, we see some variation in the patterns of selection within populations. And this is something that you might pick up if you have a longitudinal study in different habitats. You might then also compare the different types of habitats and see that there is some variation in selection. If we add these two up, right, then what we actually see is that 60% of the variation in fluctuating selection is due to spatial effects. So there is lots of spatial variation, the habitat differences that account and select for different types of personalities. Temporal variation also explains some variation, and this is of course the variation that we know most about in the literature, because most of us, for example, if you're doing your PhD for 3 or 4 years, you might measure the patterns of selection in 2 or 3 years and report that in a paper that then comes into your PhD thesis, right? So this we know a lot about. Just to show you very briefly, we're getting towards the end of the presentation actually. So we know quite a bit about what is causing this variation in selection pressures over time within single population. This is just an illustration, this work by Marion Nicolaus, who is at the University of Groningen. She showed that this variation in the patterns of selection from favoring fast to favoring slow explorers actually relates to the social environment very much, to the breeding density, for example. And this is what we can see here, that in years where the densities are increasing, selection changes and becomes more negative. So then the slower, more timid, less aggressive types do better. But in years when the breeding density goes down, then we actually see selection for the faster types. So we know a little bit about that. This is not actually the norm as far as we know as well. So this is not something that characterizes all these graded populations, but this is an example of temporal variation that is related and explainable by things that we have been able to measure. Okay. Now then we also have interaction patterns, and that of course means that that there is habitat variation, and the habitat actually interacts with year effects to explain patterns of variation in selection. It doesn't explain so much variation, but this is nice. Here we now see a nice partitioning of the variation that really shows us that it's important to consider the evolution of these types of behaviors at a larger scale. If we wouldn't have done that, we wouldn't have actually understood how important spatial variation in selection actually was in this system. Okay, does this help understand why these types coexist? Why there are these types? Yes or no, maybe. Yes, in the sense that these patterns of macrospatial variation, they would typically select for local adaptation. At the same time, we know that patterns of temporal variation counteract the speed of local adaptation. And as I said, in species with overlapping generations like the gratids, this is said to also contribute to the maintenance. But one important ingredient here that we should study now in the future is this notion that the types are also, different types of animals are also very dispersive and different their dispersal. We know this is a highly dispersive species. So this is just sort of exemplified by this little ring that I've put here. Most animals, of course, breed relatively near home, but lots of animals, particularly females, and particularly the bolder explorative females, they breed further from home. So what we think is going on is that we have sort of different adaptive peaks, basically in space and maybe also in time. They might actually select for the reduction and loss of this variation in personality, but because we have a lot of dispersion in the system, we might, because of that, also maintain this variation over evolutionary time. But there are lots of open questions about who disperses and why. Do animals select a habitat? Do they actually go to places that are well for them, and so on and so forth? Okay, brief summary then. What we have learned for the gratids, and this is also true for any other taxa that people have studied, to be honest, the gratids were really just an illustration. Individuality in behavior is the norm, is not the exception, is the norm. As far as we can see, this variation is not noise around an adaptive mean. That has very long been the paradigm. That, of course, there's some variation in our experiments, our behavior experiments. This is sort of just random noise. If it's not random, it doesn't really matter. We think it matters because, as we've seen today, it, for example, affects how well you do in different types of conditions. So this is not simple noise around an adaptive mean. We know this because there's very strong evidence for heterogeneous selection, also from many other studies, from work on fish, for example, from works on insects, from works on lizards. What causes this variation, the patterns of selection, is something that requires experiments. I've shown some of them today, actually. Population density, or in a broader sense, competitive regimes, are probably very important in generating and helping maintain this variation in these behavioral strategies. Predation risk is probably also important because what these animals differ in primarily is their willingness to take risk. And this, of course, can get you killed, and therefore cost benefits vary with predation risk. And ultimately, probably this relates to aspects of the habitat, because the habitat will offer food and therefore attract individuals and predation. Now, where we're going next really is to move away a little bit from this idea of that we have like causes and consequences. And so what we've seen is that there is this individual variation, and maybe under some environmental conditions, certain types do better, in other conditions, other types do better. But what we really think is partly because we have these types, variation is then maintained. So the idea would be that the environment is heterogeneous, not just in terms of all kinds of biotic factors like food and predators, but also heterogeneous in terms of the types of personalities that are around you. And that is probably an important pattern of selection. And so if you are interested in thinking a bit about it, read up on some of the quantitative genetics literature on indirect genetic effects, social selection, and social evolution. That is an area that we are now going to be very interested in the next coming years. And so the idea here is that these diverse personalities, they might result from certain patterns of selection, but they also induce patterns of selection that might help us understand why these types exist at all. And all of this requires long-term, longitudinal studies in the wild. And this is why I think it is great that you guys have this series to promote longitudinal field studies. It's hard to do this, I should say, because it's hard to acquire continued support from funding agencies to do such studies. But I should also say it is insightful and it's very rewarding. Okay, so with that, I would like to stop. There are many people to thank, funding agencies, lots of postdocs, PhD students, TAs, and of course, many bachelor and master students that helped us across the years, and of course, many collaborators. And with that, I'd like to end. Thank you for your attention.
Great, thank you so much. That was a fantastic talk. I think I'm going to start us off with questions while I encourage everyone in the audience to please submit something via the Q&A, unless you want to just listen to Matthew and I ask questions, which is totally fine. I'm curious a little bit more about the indirect genetic effects work that you're currently doing, and I'm curious if you could speculate on if you included those in your overall selection analyses, do you think you'd have a different story?
Yeah. I think I'll just stop the share screening, okay? Yeah. I think yes, right? These indirect genetic effects, they are intriguing. They're very hard to get your head around, but you can actually, okay? So it's worthwhile to think about that. One way by which indirect genetic effects are important is that they affect the amount of heritable variation that's available to selection. So in our crickets, for example, we are finding that aggressiveness is heritable. So crickets, right, field crickets, they're very suitable for even people do games with them, right? So you shake them, you can shake them, reset them, and then you can let them go like dices, and you can test you can have them compete, and these are people do that for gambling, right? In Eastern Asia, for example. So we use these crickets to then do stage fights, and then we have animals rotating in different diets, right? I fight with you, and with the third one, and the fourth one, and so on, right? And then you can actually estimate an individual's merit. So how aggressive is that individual in the average social environment? But you can also test how an individual, how much aggressiveness it is eliciting in someone else, right? So how aggressive you make others, that can also be repeatable and heritable. And those two can be generically correlated. And what we find in the crickets is that crickets that have genes that make them aggressive also have the genes for depressing aggressiveness in others. So there's a negative genetic correlation. Now, if you then calculate the total heritability in the behavior, and what you're actually seeing is that if you acknowledge the existence of these indirect effect and the covariance between the between the direct and the indirect effect, the heritability goes down to 0. So that means you can try to push the system by imposing lots of selection, but there will be no evolution, right? And so in the beginning of my talk, I talked about sort of the genetic architecture, right? And basically this is a component of the genetic architecture, like the behavior that individuals express in the social context and how they also affect others in that social context. And these genetic correlations, they're going to be of prime importance. So I think that will be very important. Also, I think we can very well imagine that that there are patterns of selection that you can reveal by really going into these indirect effects. And one of them is, of course, social selection, right? That the phenotype of your neighbor is going to affect your phenotype and thereby your fitness. And for example, through negative frequency dependent selection, but that can also be through other roles. And so there's a lot to explore and learn. It's hard to do, I should say, in the wild. It's very hard.
Yeah. Good luck. I'm interested. All right, Matthew also has a question. I'll let him take it away.
Yeah. So my question has 2 parts. The first part, so with you showed all of that variation in the strength of the selection coefficient and the direction of the selection coefficient, and variation both across years, within sites, across sites. And so it would seem like if birds can match their phenotype to the ideal, that would be good for them. I'm guessing they don't do that though. Is that right?
Yeah. No, this is exactly the problem, right? So if you think back about this density dependence, right? So when the density goes up, slower birds do better. So what should you do? You should just become slower, right? And we know that animals are super plastic. So that's what they should do. There is actually then selection for reversible plasticity. That's one way to see it, right? So the environment changes, you should change with it in a particular way. For that study, we looked at that actually. We quantified the extent to which individuals change their behavior in relation to density and whether they actually become slower. They don't. So in that study, we were finding that yes, these individuals are repeatable in their behavior. That behavior in that study was also heritable. Yes, there is density-dependent selection. No, they're not actually modifying their behavior with density. And so the question is, why is that, right? And then we are, of course, we have to think about some of the costs and limits to plasticity. So even though we think, oh, behavior is super flexible, should be possible to just change our behavior, we are, of course, talking about some hard wiring, right, that we might need to change, right? The types of neurons that are connected in certain ways and fire in certain ways, right? So it might not be that easy, actually, to always match the environment that you're experiencing. I mean, there are no Darwinian demons, right? And there could be, on the one hand, there could be this sort of cost of plasticity, that it's just costly to change somehow. But there are also other types of costs, right? If the environment is changing, you need to perceive this change. And that is, of course, also very costly. Now, there is in this context some interesting work that also tries to understand why the animals have personalities. That implies that there are feedback loops. And the idea here would be that that if you, once you've shown a particular type of behavior, the costs of doing that go down because you become more experienced, more skilled, you could say, right? And therefore, next time you face this decision of how to behave, you might behave in the same way, which might be different from another one. And then these positive feedback loops, they would actually lead to a situation where basically individuals are selecting different environments and differing in their behavior, so that at the end of the day, you end up with these different personalities that are not flexible anywhere, not plastic. Yeah, good question. This is the question, right? If patterns of selection change, why the hell do they not change? In some ways they do sometimes, but certainly that's not always the case. And this must be part of the answer.
Thanks.
Do you find one last question, do you find higher levels of reversible plasticity or any levels of reversible plasticity in those populations or plots that were really highly variable in selection between years compared to ones where they were lower?
Yeah, it's a good question. We have to look into that. The patterns of repeatability don't tell you so much about that, because the repeatabilities were very similar, but they can be similar even if the variance among individuals and within individuals is very different. And so one way to get around that, right, and also the mean values might be a little bit different, is to calculate coefficients of variation. And this is what many people have now starting to do, right? You can estimate the variance among individuals and divide it by the mean. And then you have basically an index of individual variation that you can then compare across the population. And you can also do that with the within individual variance. And that would directly address your question, right? So I think that's what we would need to do. We haven't done that, but we should. Yeah, and I know that there are now people reworking essentially these meta-analysis of repeatability to find, to use the actual estimates of within and among individual variance and the mean values to really look at whether there are patterns in reversible plasticity across the meta-analysis, right, across species or environments or contexts, as well as patterns of individuality. Yeah, that's where we're going. But we're starting with very complex statistics, and then we're making the statistic, of course, even more complicated, right? To go back to answering biological questions.
Yeah, of course, biology is complex, of course. All right, well, maybe we'll stop there with that profound insight. Thank you so much to both of our speakers today and for everyone out there listening. And just as a reminder, next week we will have a pre-recorded talk, so please check that out on YouTube, and the link will be available on Twitter. Thank you all so much, and have a great evening, afternoon, day, wherever you are. Bye.
Bye.
Bye.